This video is a follow-up to the previous video on protein domains. We have discussed that proteins are involved in virtually every biological process imaginable, acting as tiny machines that carry out specific tasks within our cells. These tasks range from transporting molecules to catalyzing chemical reactions and providing structural support. Proteins are made up of building blocks called amino acids. There are 20 different types of amino acids, each with a unique chemical structure. The specific sequence of amino acids determines a protein's three-dimensional shape and ultimately its function. The intricate folding and interactions between these amino acid chains create specialized regions called domains. These structures impart on a protein the ability to perform specific functions. Understanding protein domains is crucial for unraveling the complexities of protein interactions. In the following sections, we will delve into three important protein domains: SH2, Zinc Leucine Zipper, and ASH domains. One of the main means of cell-to-cell communication is by a process called signal transduction. The first messenger, which is usually a hormone, itself a protein, attaches to a receptor on the cell membrane. This attachment initiates a cascade of reactions which usually involves transcription factors that can influence gene expression. SH2 domains, short for SRC homology, 2 domains are present in signal transduction proteins. They are around 100 amino acids in length and are similar to regions of tyrosine kinase enzymes and have binding regions for phosphorylated tyrosine residues present on other proteins. This allows them to link a variety of tyrosine kinases to molecules further along the reaction cascade. The importance of these SH2 domains can be gauged by the fact that there are 120 present in some 110 proteins in the human body. These proteins include kinase enzymes, which attach phosphate groups to molecules, an important switch in regulation of cascade reactions, and phosphatase enzymes, which reverse the action of kinases by removing phosphate groups from molecules. These factors highlight the importance of SH2 domain attachments in initiating a cascade of downstream signaling events. For example, when a growth factor binds to its receptor on the cell surface, it can trigger the phosphorylation of tyrosine residues on the receptor itself. This creates binding sites for SH2 domain-containing proteins, leading to their recruitment and activation. Considering the presence of SH2 domains in a wide variety of proteins involved in cell signaling, including enzymes, adapter proteins, and transcription factors, it is not surprising that mutations in SH2 domains can disrupt these signaling pathways, contributing to diseases like cancer. DNA, the blueprint of life, contains all the genetic information necessary for an organism's development and function. Regulating which genes are expressed and when is crucial for proper cellular function. Zinc Leucine Zipper domains play a critical role in this regulation by binding to specific DNA sequences. This domain has a coiled coil structure formed by 2 alpha helices. It is made up of a zinc finger motif binding to a zinc ion. This region aids DNA binding, whilst the leucine zipper motif, which contains a repeating leucine residue every 7th position. These leucine amino acids interact, zipping the helices together, facilitating protein dimerization. The zinc ion stabilizes the structure. An example of a protein containing the zinc leucine zipper domain is ZIF268. It is, like all such proteins, a transcription factor which may play a role in memory formation. This is based on the fact that its mRNA is upregulated during associative learning and long-term potentiation, LTP. It can be fused to the leucine zipper of the protein, GCN4 in yeast, to create a dimeric zinc finger protein. ASH, which stands for SPMSPD2 hidden domains, act as the architects of chromatin structure. The very long DNA molecule is intricately packaged with the aid of histone proteins into chromatin. The ASH domain allows proteins to bind to specific histone modifications, particularly methylation marks. By binding, ASH domains recruit proteins involved in chromatin remodeling. This packaging regulates gene expression by controlling DNA accessibility. The ASH domain is also present in proteins associated with centrosomal and ciliary structures, as well as the Golgi complex. So let's review the significance of domain-domain interactions in different cell signaling processes. we have explored three distinct protein domains: SH2, Zinc Leucine Zipper, and ASH domains. These domains play diverse roles in cellular processes. SH2 domains mediate cell signaling, Zinc Leucine Zipper domains control gene expression, and ASH domains contribute to chromatin remodeling. These interactions are crucial for cellular function. Therefore, their disruptions can lead to disease, which means understanding them is critical in unraveling biological complexities. The greater our understanding, the greater the number of potential therapy opportunities. We will provide updates on new breakthroughs achieved by the research teams in all corners of the globe. If you like this video, don't forget to hit the like button and subscribe to this channel. Also hit the notifications bell to be alerted to new videos published. And of course, stay curious.